Welcome to the deep dive. Today, we're really getting into the nitty-gritty, the fundamental chemistry that makes life tick.
That's right.
Our mission really is to understand how these essential building blocks inside us work, from water right up to the really complex stuff. We're doing a deep dive into the chemical components of a cell, exploring how tiny atomic interactions make everything happen. And there are some fascinating things we'll uncover.
Oh, definitely.
Like how atoms are always seeking stability, how bonds actually store energy, and why water, which we totally take for granted, is just so perfectly suited for life.
And what's really illuminating, I think, is that these principles, we're talking about atoms, bonds, interactions, they're truly the bedrock. Everything in biology builds on this. Pretty much. From the smallest reaction to the biggest protein, it all comes back to these basic chemical rules. So this dive should give you a much clearer picture of how cells get built from carbon compounds, how they have these families of small organic molecules, and ultimately how they're dominated by these huge macromolecules with amazing properties. It's all down to the chemistry.
Okay, let's start at the very beginning then. To understand how all that cellular machinery works, that incredible precision, we need to go back to basics. Atoms. What drives them to interact? Is it just random bumping?
Not quite random, no. It really boils down to stability. We know atoms have the nucleus, protons, neutrons, and orbiting electrons, right?
Right.
But the key is those electrons aren't just whizzing about anywhere. They occupy specific energy levels, or shells. Think of them like layers.
Okay.
And the outermost shell, the valence shell, that's the crucial one. Atoms are, let's say, happiest or most stable when that outer shell is completely full.
Like having a full set of something.
Exactly. So if an atom has a partially filled outer shell, it's inherently less stable. And this isn't just a concept, it's a driving force. This instability makes them want to react.
So they're actively trying to fix that.
Precisely. They're constantly trying to, reduce that instability, find a way to get a full outer shell. And the main way they do that is by interacting with another atom.
So that's the prime mover, the search for a full outer shell. And how do they actually, interact? What are the main ways?
Yeah, that drive leads to different kinds of interactions.
Yeah.
Now, there are ionic bonds where electrons are actually transferred. But for life inside a cell, we're mostly focusing on two big ones, covalent bonds and hydrogen bonds.
Got it. Covalent and hydrogen bonds. Let's tackle the covalent ones first. They sound pretty important. How do they work?
They are incredibly important. So covalent bonding is all about sharing. Imagine two atoms. Neither has a full outer shell.
Okay.
They can essentially share one or more electrons from their outer shells. By sharing, both atoms kind of feel like they have a full shell, or at least a more stable arrangement.
Like pooling resources.
Exactly like that. And this sharing forms a covalent bond, creating a new, stable unit, a molecule. These bonds are generally very strong and stable. They form the actual backbone, the skeleton, of most biological molecules. Think of them as the strong rivets holding things together.
Strong and stable. But I remember reading that even these strong bonds aren't all identical, are they? There are differences.
That's a really key point. Yes, they come in different types, and those differences matter hugely for how molecules behave. You've got single bonds sharing one pair of electrons. These are pretty flexible.
Flexible how?
The atoms can sort of rotate around the bond axis like swiveling. This gives parts of molecules freedom to move.
Okay, I see.
Then you have double bonds. Here they share two pairs of electrons. These are shorter, stronger, and much more rigid. No swiveling allowed, really.
Rigid. Why is that important?
Oh, it's critical. Think about fitting molecules together precisely, like an enzyme needing to hold its target just right, or parts of DNA needing to be flat. That rigidity is essential. There are also triple bonds, even stronger, but they're less common in biological molecules. Nitrogen gas in the air, that's a triple bond.
So the flexibility from single bonds and the rigidity from double bonds that directly controls the shape and function of big molecules like proteins.
Absolutely. That flexibility lets protein chains fold into incredibly intricate 3D shapes, which is vital for their job.
Right.
And the rigidity of double bonds helps maintain specific structures, like keeping the base pairs in DNA flat and stacked correctly. It's these details, these specific types of strong connections that determine so much.
Okay, this is where my mind started racing when I was reading up. Covalent bonds aren't just about structure, they're about energy too, right? You mentioned strength. How does that relate to energy stored in the bond?
Yes, exactly.
Yeah.
This is fundamental to, all of metabolism.
Yeah.
The strength of a bond is related to the energy difference between the atoms when they're separate versus when they're bonded in the molecule.
Okay.
So stronger bonds are more stable. think of them as being in a lower energy state. They don't have much extra energy to give away easily.
Huh.
Molecules like CO2 or water have very strong, stable bonds.
So not much fuel there for a cell.
Not much readily available fuel, no. But now, flip that. Weaker covalent bonds. They're less stable. And because they're less stable, they possess more potential energy that can be released relatively easily when the bond breaks.
Ah, so weaker bonds mean more stored energy. That feels counterintuitive at first.
It does, but it's key. Think about sugars, carbohydrates. They have lots of carbon-hydrogen bonds, which are comparatively weaker, less stable.
Like CH2O you mentioned.
Right, the basic sugar unit. Those bonds hold energy that the cell can tap into. That's literally why we eat food containing sugars and fats. They're packed with these energy-rich, weaker covalent bonds.
Wow. Okay, so the cell needs that stored energy constantly for moving, building things, transport.
Everything. Absolutely everything. Understanding which molecules have these higher energy bonds is central to understanding how cells get their power. It's the energy currency.
And what causes these differences in bond strength and energy? You mentioned something about atoms attracting electrons differently.
Yes, that's electronegativity. It's basically a measure of how strongly an atom pulls shared electrons towards itself in a covalent bond.
Okay.
Some atoms, like oxygen or nitrogen, are highly electronegative. They pull electrons strongly. Others, like hydrogen or carbon, are less so.
And if you bond atoms with different electronegativities,
Then the sharing isn't equal. The electrons spend more time closer to the more electronegative atom. This creates a slight negative charge near that atom and a slight positive charge near the less electronegative one.
oh, the charge isn't spread evenly across the whole molecule.
Exactly. You get an uneven distribution of charge. We call that a polar molecule. It has positive and negative poles, so to speak. And these polar molecules are incredibly important in biology.
Because that polarity allows for other kinds of interactions.
Precisely. That slight charge separation, that polarity, it opens the door to another crucial, though much weaker, type of bond.
Okay, so we have these polar molecules, slight charge differences.
Yeah.
What happens next? You mentioned another type of bond earlier.
Yes, the hydrogen bond. This happens when you have these polar molecules interacting. Specifically, it's an attraction between a hydrogen atom that's already part of a polar covalent bond.
So it has a slight positive charge.
Right. An attraction between that slightly positive hydrogen and a strongly electronegative atom, usually oxygen or nitrogen, on a different polar molecule, which has a slight negative charge.
So it's like a weak magnet, an attraction between these slightly charged parts of different molecules.
That's a good way to think of it. An electrostatic attraction. Now, here's the thing. Individually, a single hydrogen bond is quite weak, maybe only about 1/20th the strength of a covalent bond.
Pretty flimsy on its own, then.
On its own, yes. But, and this is critical in biological systems, you rarely have just one. You often have hundreds, thousands, even millions of them working together. And collectively, they become incredibly significant.
Ah, strength in numbers, like lots of tiny threads making a strong rope.
Exactly. Think about water. H2O, oxygen is very electronegative, hydrogen less so. So water is highly polar.
Right.
And water molecules form hydrogen bonds with each other like crazy. That collective hydrogen bonding is responsible for nearly all of water's amazing life-sustaining properties.
Like what specifically?
Well, its high boiling point, for one. It takes a lot of energy to break all those hydrogen bonds and turn liquid water into gas, which keeps water liquid over a temperature range suitable for life.
Yeah, okay, that makes sense.
Its ability to dissolve so many other polar substances, it's the universal solvent, right? That depends on hydrogen bonding. How it sticks together, cohesion, sticks to surfaces, adhesion, vital for plants moving water up stems. All down to hydrogen bonds.
Wow. So these supposedly weak interactions are actually fundamental.
Absolutely fundamental. They're not just some minor effect. These hydrogen bonds and other similar weak interactions allow for the dynamic, constantly changing, yet stable structures we see in biology. Think protein folding.
How a protein gets its specific 3D shape.
Exactly. Hydrogen bonds play a huge role in holding that shape. Or think about DNA. The two strands of the double helix are held together primarily by hydrogen bonds between the base pairs.
Strong enough to hold it together, but weak enough to be unzipped when needed, like for copying DNA.
Precisely. That's the beauty of it. They provide stability, but also flexibility and reversibility. If bonds were all super strong covalent bonds, everything would be locked solid. If they were too weak, things would fall apart. Life operates in that sweet spot, using a combination of strong covalent bonds for the framework and numerous weak bonds, like hydrogen bonds, for dynamic structure and interactions.
That balance seems key. So let's recap a bit. We've gone from the atom's basic need for stability,
Filling those outer electron shells.
right, through the strong covalent bonds that form molecules, remembering that single bonds offer flexibility and double bonds rigidity.
Which dictates shape and function.
And we learned that bond strength relates to stored energy weaker bonds, actually holding more accessible energy for the cell,
The fuel of life.
driven by differences in electronegativity, which creates polar molecules. And finally, these polar molecules interact via individually weak but collectively powerful hydrogen bonds. And these hydrogen bonds give water its unique properties and are crucial for structures like proteins and DNA. It's kind of mind-blowing how these simple chemical rules build everything.
It really is elegant, isn't it? The efficiency of it all. This chemistry truly underpins everything a cell does. Signaling, building, energy, communication. It's all molecular interactions governed by these principles. Understanding this is like having the architectural plans for life itself.
Amazing.
Now, here's something to really think about. We talked about that balance, the collective strength of weak bonds like hydrogen bonds provides stability, but also allows for constant change, rearrangement, dynamism. How does that very balance, that interplay between holding things together just strongly enough, but not too strongly, allow life not just to exist, but to adapt, respond, and thrive in a world that's always changing?
That's a deep one. A perfect balance for a dynamic existence. Thank you for joining us on this deep dive into the chemical foundations of the cell. Keep exploring, keep questioning, and we'll connect with you next time.